diabetes mellitus. Diabetes is a chronic metabolic disorder characterized by high levels of glucose in the blood. This condition arises due to either insufficient insulin production, ineffective use of insulin, or a combination of both. Insulin is a hormone produced by the pancreas that helps regulate blood sugar levels by facilitating the uptake of glucose into cells for energy production or storage. Diabetes is one of the leading causes of death in the United States. About 38 million US adults have diabetes, and one in five don't know they have it. Diabetes is the number one cause of kidney failure, lower limb amputations, and adult blindness, and in the last 20 years, the number of adults diagnosed with diabetes has more than doubled. Type 1 diabetes results from the autoimmune destruction of insulin-producing beta cells in the pancreas. As a result, the body produces little to no insulin. Type 1 diabetes typically develops early in life, often during childhood or adolescence, and requires lifelong insulin therapy for management. Type 2 diabetes is characterized by insulin resistance, where cells become less responsive to insulin's actions. Initially, the pancreas compensates by producing more insulin, but over time, insulin production may decrease. Type 2 diabetes is more common than type 1 and is often associated with lifestyle factors. While it can occur at any age, it typically develops in adulthood. Initial symptoms of diabetes include increased thirst, fatigue, frequent urination, blurred vision, unexplained weight loss, and slow healing cuts and sores. Prolonged exposure to high levels of glucose in the blood can lead to serious complications from diabetes, including nerve damage, heart disease, and kidney disease. Insulin is a hormone produced in the pancreas which regulates the amount of glucose in the blood. Insulin helps blood sugar enter the body cells so it can be used for energy. Insulin also signals the liver to store blood sugar for later use. The human insulin protein is composed of 51 amino acids, and it is a heterodimer composed of an alpha chain and a beta chain, linked by two disulfide bridges. Insulin is a peptide. Peptides are short chains of amino acids linked together by peptide bonds. Insulin is composed of two polypeptide chains, an alpha and a beta linked by disulfide bonds. Let's look at how insulin is produced in the pancreas. So after a meal, blood glucose levels naturally rise. Beta cells in the pancreas detect glucose in the blood and release insulin right back into the blood in order to trigger the import of glucose into cells. Located on the surface membrane of pancreatic beta cells are glucose transporters, GLUT2. Glucose transporters help import glucose into the beta cell. Through glycolysis, the citric acid cycle, and oxidative phosphorylation, you produce ATP. The level of ATP in the cell is directly proportional to the level of glucose in the cell. So the more glucose we have in the blood, the more ATP is produced. Elevated ATP levels inhibit the potassium channel. This stops the potassium efflux and triggers the depolarization along the membrane of the beta cell, which activates the voltage-dependent calcium channel, leading to increased potassium in the beta cell. When there is elevated blood glucose, we have increased calcium in the cell. That calcium will then trigger insulin exocytosis, leading to insulin being secreted right back into the blood. The whole process is a graded response. meaning that the more glucose in the body, the more ATP produced, and therefore the more calcium in the beta cell, and thus more insulin being released right back into the bloodstream. Now let's look at the insulin receptor protein. The insulin receptor is a type of transmembrane receptor found on the surface of many cell types throughout the body. When linked with insulin, it plays a crucial role in regulating glucose metabolism and other metabolic processes. The structure of the insulin receptor ectodomain is saturated with 4 insulin ligands and reveals a tea-like or bouquet of flowers-like conformation with converging membrane proximal domains and structural evidence of insulin binding sites. The insulin receptor is a glycosylated disulfide-linked alpha-beta 2 homodimer with a modular domain structure. Each protomer consists of an extracellular linkage binding with an alpha-beta subunit and the membrane-spanning beta subunit. We also observe 2 distinct binding sites. 2 of the observed insulin binding sites are located in the top or head region and the additional 2 insulin molecules are located in the bottom or stalk region. Here we see a close-up of binding sites 1 and 2. In site 1, we observed several functional groups, including glycine, isoleucine, valine, glutamine, tyrosine, asparagine, valine, tyrosine, glycine, phenylamine, tyrosine, and insulin residues. In site 2, we observe serine, leucine, glycine, histidine, glutamic acid, and leucine. Here we see more close-up views of the 4 insulin and their receptor binding sites. Insulin, colored red, binds to the alpha subunit of the insulin receptor protein, which is colored blue. The insulin receptor spans the cell membrane, so it has a part inside and outside the cell. This binding activates the cytoplasmic beta subunit of the insulin receptor, RTK. It is part of the beta subunit of the receptor. The insulin receptor has 2 components, and they initially start off being separated. When insulin comes and binds to the insulin receptor, it makes both receptors combine to form a dimer. These 2 protein receptors activate each other, and this activation allows signals to enter the cell and specialized proteins interact with the receptor. The receptor activates these proteins and they in turn activate other proteins in what is a large, complex cascade that results in the cell taking up glucose for use as an energy source. In people with type 2 diabetes, insulin binds to the insulin receptor, but this does not lead to a signal cascade. Over time, high glucose levels damage many different organs. Lastly, we'll look at treatment options for people with diabetes. Insulin glargine is a man-made product that is similar to human insulin. It replaces the insulin that your body would normally make. It acts longer than regular insulin and provides a low, steady level of insulin. It works by helping blood glucose get into the cells of your body that you can use for energy through the pathways described earlier before. Interestingly enough, insulin used to be made from pigs, as it is close to the human version, as it differs only by the C-terminal amino acid of the beta chain. In humans, that amino acid is threonine, and in pigs, it's alanine. Ultimately, treatment and management of diabetes involves lifestyle modifications such as diet and exercise, monitoring blood sugar levels, and in some cases, medication. The goal of treatment is to keep blood sugar levels low within a target range to prevent complications and maintain overall health and well-being. Thank you and have a good day. References.